I start into my presentation, I would like to convey gratitude to the members of the Bukhari Knowledge Fest 2020. And it's a great privilege to be a part of this festival. And I'm equally excited to present a talk in front of such a diverse audience. Hence, I would try to make my presentation, my talk, as much informative as possible. So before I go into my presentation, I would like to give a brief description about my educational background. So I have attained my PhD in environmental toxicology from India, and over a years of research, I use various theoretical and experimental models to understand how chemicals interact with living beings. Or in other words, let me say my area of research is to understand the effect of chemicals in human health. So if you look around, you would see that you are surrounded with chemicals. We as human beings practically and literally live in the world of chemicals. Chemicals are present in our life and in daily life. For example, when you wake up in the morning, the toothpaste which you use is actually contains hundreds of chemicals. The coffee mug in which you had your coffee is actually a chemical product. The toy which you have bought for your kids is actually made up of hundreds of chemicals like plastic, plasticizers, emerging pollutant, and so on. Even there are chemicals in your peanut butter. So we can understand that we are actually living in the world of chemicals. So now this brings us to a very important question, which is how safe are these chemicals? Or in other words, are these chemicals which we are using has any side effect for us or not? The answer to this question might surprise you. A recent study has found out that only 3% of the chemicals which are present in our environment has been thoroughly and comprehensively tested, whereas the rest 97% of the chemicals has been barely or minimally tested. Let me give you another example that might be an eye-opener to you. It is estimated that there are something like 190,000 chemicals present in our consumer products. And these chemicals have been superficially tested or not tested at all. When I say about consumer products, what does it mean? Consumer product means anything which you use daily. For example, a shampoo, body lotion, your popcorn bag, anything which you use regularly. And what does the word mean comprehensively tested? Whenever there is a chemical which is subjected for human use, the chemical has to be tested through a lot of endpoints, which means that whether this chemical is a toxicant to neurological behavior, whether this chemical is a liver toxicant, whether it damages your kidney, whether it damages your skin. So likewise, there are a lot of tests for which a chemical has to pass. And if the chemical passes, then only we say it has been comprehensively tested. So only 3% of the chemicals, which actually includes drugs, pesticides, and food products, have been only thoroughly and comprehensively tested. The rest 97% of the chemicals have been superficially tested. Now this brings us to another question, which is a very relevant question. The question is actually, where are we failing? Why the scientific fraternity fails to conduct the risk assessment for this 97% of the chemicals? Why as a society we are being kept or we are being exposed to such huge number of chemicals for which we have no data or no idea? The answer to this question might again take you by surprise, but the answer to this question is actually the problem or the manner in which we conduct risk assessment of chemicals. And traditionally, to evaluate the effect of a chemical, animal experimentations are employed. Animals of different species are used to understand the chemical safety, and this is where lies the problem. Chemical testing using animal is actually more of a problem than it's a solution. And I would tell you why animal testing is actually a problem. First reason for which the animal testing is a problem is animal tests are poor predictor to human response. What does that mean? It means that whatever data or responses which we obtain in animals are either not fully extrapolated or either not fully translated to humans. And why is that so? A simple answer to this is a rabbit is not a 70 kg human. Though scientists for long have said that humans and animals share genetic similarity, molecular physiology, but still animal experiments fail to replicate the results in humans. I will cite you an example. Rats and humans have 96% genetically similar, but still there are a lot of experiments, a lot of chemicals which have been shown to be effective in animals are actually proven to be detrimental to humans. Why? Because though 96% we share similarity with rats, but the 4% indifference makes all the kind of problem for chemical safety. For example, there was a drug named thalidomide. This drug was given to pregnant women as a sedative, and the pregnant women, they used to pop the pill in the morning so as to alleviate themselves from morning sickness, nausea, and others. This drug was considered to be a wonder drug during the 1980s. Surprisingly, babies born out of this pregnant woman were either born crippled or were had no limbs present with them. Interestingly, this drug thalidomide has passed all the animal tests, and the chemical was labeled as safe. So now you can understand, and this is not just one chemical. There are hundreds of drugs which has been removed from the market because of unseen side effects. So that is the reason why this is one of the reason of animal disadvantage in safety evaluation of chemical. Second thing, animal tests are not reliable. What does that mean? It means that when we do testing of a chemical, we use different species. For example, we use rat, we use mice, we use rabbit, and the genetic physiology of these species are very different from each other. And this is altogether different from humans. Like, for example, the body temperature of human is quite different from a bird or another animal. The sleeping pattern is different. So likewise, there is different physiological aspect between the species. This is called a species difference. And because a drug is given to the species, each species react differently to each drug. And that is the reason why the drug industry at now is struggling and spending so much money to come up with a drug, with a safety drug. And that is one, for example, I would tell you animals, another reason for animal experiments not to be successful or not to be as successful as it thought to be is because animals naturally do not suffer from the diseases of civilization. Animals do not have diabetes, they do not go through heart attack, or they do not show symptoms of hypertension, but yet we use animals to find the effect of such drugs. And in doing so, what we do is that we make the animal sick. Diabetes in rats is caused by giving rats high doses of poison so that their liver cells are damaged and destroyed. Then scientists administer anti-diabetic drugs to find out how safe and effective this anti-diabetic drug is. Heart attack is caused by constricting the arteries in animals. In the same manner, there are animals which are purposefully bred to mimic certain kinds of disease. Let me give you another example in simple words. Out of 10 drugs, only one drug reaches to your pharmacy shelves. Rest of the 9 drugs which passes animal test fails in human clinical trials, and only one drug reaches your market. The success rate of a drug development is 0.02%, and this is because the drug development is highly relying on the data obtained from animal experiments. So the second disadvantage is that animal experiments are non-reliable. Third disadvantage is data generated out of animal experiments are misleading. Animal experiments, we sometimes see false positive and false negative, and because of which we think that that particular drug or chemical is not effective in humans. And this is where lies the problem. For example, penicillin was found to be highly disastrous to rats and mice. But as of now, penicillin is the world's first naturally discovering antibiotic, and it is the most widely prescribed antibiotic drug in the world. Second example I can cite you is named as tamoxifen. Tamoxifen is a breast cancer drug. It is considered as a blockbuster drug to treat women suffering from breast cancer. Breast cancer initially was shown to cause liver tumors in rats, but for human it is considered as one of the best drugs ever developed to treat breast cancer. So we can understand that animal experiments are misleading. Their results can hurt humans. The fourth and the most important point is animal experiments are very costly and it takes huge time. If you see, if I'll cite you the case of one case, which is drug industry. If you see in drug industry, it takes $20 million to develop one drug. So if you have one compound, if you are going to develop that compound into a drug, it takes $20 million. And on top of that, if it is found to be effective, then it will take 10 to 12 years for that particular compound to reach your market as a drug. So you can understand how much the time it takes. The last point is that animal experiments are extremely cruel and inhumane. Animals are subjected to severe pain, distress, and trauma during experimentation. They are fed not properly. They are given high doses of medicine so that they develop disease. They are caged, they are gagged, they are given poison. So these are all kind of inflictment that humans do on animals. One such example is the use of animals for development of cosmetics. If you look closely, cosmetics are actually not a necessity or need for human beings. Cosmetics is actually a luxury. It is not a necessity. So for human luxury, hundreds and thousands of rabbits, rats, mice are killed. I'll tell you one specific example which will make you frightened or shocked is that the eyeliner which girls apply, the process of safety evaluation is how brutal, I will tell you. Generally, rabbits are used for safety evaluation of the ingredients used in eyeliner. The rabbits are held by their neck, their eyes are stretched wide apart using a mechanical instrument, and the chemical of interest is dropped on the eyes of the rabbit. If the rabbit eye bleeds, it means the chemical is not safe. If it doesn't bleed, if there is no anatomical or physiological changes in the eyes of the rabbit, the chemical is labeled safe and it can be used as an eyeliner. Can you imagine the pain and distress an animal goes through for the luxury of humans? Another example, animals don't smoke, but to understand the effect of smoking, scientists expose rat to a stream of tobacco smoke, and this happens for over months and months. And finally, to see the effect of smoking on different organs of the animals are killed and the organs are extracted. Animals are subjected to electric shock in order to study depression. Furthermore, animals don't drink, but in order to understand how alcohol makes a neurobehavioral changes, a baby rat is injected with alcohol in their belly, and then it is monitored to understand whether alcohol has any neurobehavioral changes in young adults. These are some of the limitations of animal experiments. So, what we understand is that animal experiments are highly cruel, inhumane, ethically questionable. It's highly time taking, very highly costly, and most of all, the results or data which is obtained is actually not applicable to human beings. So, what is the way out? Fortunately, we are living in the world of scientific advancement, and there are forward thinking scientists and communities who have come up with path breaking and innovative ideas to develop non animal methods. We call these non animal methods as alternatives to animal testing. So, what are these alternatives to animal testing? Alternatives to animal testing includes novel innovations such as use of human cell culture. Instead of using rat, rabbit, or mice of a different species, let's use cells of humans. That is called cell culture technique. Second alternative method, which is considered as a very promising model, is called organ on chip. It is a more advanced version of cell culture. Use of computer modeling is again considered as an alternative to animal experiments. So I would like to expand upon in brief what are these alternative methods. One such method is organ on chip. Organ on chip is actually a chemical which has been a methodology developed on microfluidics technology. So now this organ on chip is actually it's actually a plastic box which is the size of your double A battery or a pencil battery. Now this double A battery actually mimics your entire organ. For example, it can mimic what happens in your liver or lung or kidney. And how do scientists achieve that? Now, scientists have made small holes in these small plastic boxes, and each hole consists of one cell of your lung. Suppose if your lung has 5 different cell types, so the plastic box will have 5 different holes, and each hole will have its one single cell type. And these cells will be connected with each other through a fluid. Now that fluid will represent blood. And it has been found that using organ on chip is more accurate and more predictive in determining the safety of a chemical than an animal test. Second innovation in the field of alternative is the use of human reconstructed skin. And this technology is considered as a groundbreaking technology to test new entities in cosmetic products, in cosmetic industry. What actually happens is the scientists have developed a method in which they use human skin cell, and this human skin cell is grown artificially in such an environment that it actually replicates the exact human skin cell. And from where do the scientists obtain the skin cell? They generally obtain the skin cell from some skin surgery or from circumcision, the skin which is left over, which is going to be discarded. That skin is taken, brought to the lab, processed, and then on top of that, what happens is that any chemical entity which is meant to be used in a cosmetic product is applied, and then the result which we obtain is far more better and accurate and predictive than using animal models. Another example of innovation is the use of computer modeling. We are now living in a world of artificial intelligence. So scientists have smartly utilized the knowledge of artificial intelligence, big data, computer modeling, and to correctly evaluate the effect of an unknown chemical. Recently, researchers from Johns Hopkins University, USA, has developed a computer algorithm. Now, this algorithm has conducted huge number of tests, and it has been found that the prediction rate using this algorithm is higher than prediction rate using animals. So what do we understand? It understands that artificial intelligence and computer modeling outweighs the benefits what we get from an animal experiments. In fact, use of robotic technology is another option. When we do manual testing, humans have a capacity to do certain number of tests. They cannot go beyond that. But if you apply robots, they can do hundreds and thousands of experiments in one day. So that is another alternative method to animal testing. So what do we understand from animal testing? We understood from animal testing is that what do we understand from alternatives to animal testing? That it is actually the alternative to animal testing is actually cost effective, ethically and morally sound. It doesn't take much time. For example, if you have to find out whether a chemical is carcinogen or whether it causes cancer, it takes 3 years to understand whether the chemical causes carcinogen in animals. But if you use this technology, we can understand whether the chemical is carcinogen or not in half of the time which is required in animals. So animal test alternatives are time saving. It's highly cost effective as compared to animal test. You remember, for developing a drug, we need billions of dollars. But if you use this technology, we can cut down the price. And if we cut down the price, the markets will be available the drugs will be available at a cheaper rate. So it is a pocket friendly to society. So here I have given you a brief idea about the problem of the animal test, what are the alternatives. So now I have come to the end of my presentation, which is that I would like to end with a take home message. The take home message is that we are now living in the world of rapid scientific advancement. And as a scientist myself and the scientific fraternity, we should utilize the modern tools in order to make our world safer, in order to make sure that the chemicals which we are using is not going to do any harm to us. Hence, using innovation, using new technologies will definitely enable in achieving this. Thank you so much.
Good afternoon, everyone. My name is Palanivel. I would like to first thank the committee, BUKHARI Knowledge Fest, for organizing this wonderful event. And my topic, I'm going to talk about nutrients and cancer. And mainly, I will explain how nutrition promotes cancer development in humans and how are we going to prevent the cancer formation in humans. This is the topic I'm working on, and mainly I'm a medical researcher. I will talk about the food that promotes the cancer development and what are the foods that can prevent cancer development. So before going into my research topic, I would like to tell you in a simple way what is cancer. Cancer means it's a tissue growth, abnormal tissue growth. Normally, every cell in human body has a lifetime. For example, the red blood cells in our blood, the red blood cell has a lifetime of about 120 days. So at the end of 120 days, the cell should die normally. What happens when the cell becomes cancerous? The cells will not be able to die after 120 days. They start to grow, keep on growing, and they acquire a lot of nutrients. I mean, the glucose, amino acids, vitamins. They eat a lot, and they grow bigger than the normal cells, and they form a abnormal tissue growth. So this is called cancer. So similarly, all the organs in our body, brain, liver, heart, kidney, intestine, stomach, every organ has a form of tumor. So these tumors developed through some risk factors. Risk factor means some bad habits of like smoking and tobacco chewing, and so this kind of habits they can cause cancer. How they can cause cancer? Smoking tobacco cause some kind of mutations in the DNA of the cells. The DNA is nothing. The DNA basically carries the information, genetic information, to another generation. So when the DNA is mutated, they don't listen to the normal body mechanism. So they start to act completely independent of any control. They try to control their growth and their eating behavior themselves. But the normal cells, they have a very uniform and definite mechanism. They grow slower than the tumor cells. So what are the main causes of cancers? The cancer, as I said in the beginning, smoking and tobacco is one of the major reasons. And then second is diet and physical activity. Diet is more important for the health, for the normal healthy life. And nowadays the modern diets like the high fat diet, including pizza, burger, cheese, and artificial sweeteners like the preserved fruit juices, preserved frozen meats, they have a lot of contaminants, they have a lot of chemicals. They also contribute to the cancer development. So the modern lifestyle also play a major role in cancer development. So we have to be careful um, in designing our lifestyle, mainly with the food and drinks. This is also very important. And other than the food, viral infection also cause cancer. For example, human papillomavirus, it's one of the virus that causes cervical cancer in women. And other than viruses, UV radiation also causes cancer. So these are the major risk factors that can cause cancer. So all types of cancer is mainly because of the DNA mutation. Mutation is nothing. The DNA undergoes some changes. The changes is referred to or defined as mutation. Mutation is the scientific term that defines where there is a change in the DNA in our body. So these mutations may be caused by aging or exposure to chemicals. Chemicals means many people work in the chemical industry. There's those hazardous chemicals, dangerous chemicals can also cause some mutations. These mutations might lead to cancer development. And then radiations, hormones, or other factors in the body and in the environment. Over time, a number of mutations. So not single mutation can develop the cancer. There are several mutations need to be acquired by cancer cells. Then only they can form cancer, aggressive cancer. So what type of mutation causes cancer? The most commonly mutated gene is something that has an information. For example, our body, every tissue has specific roles, and this specific roles is driven by specific genes. So the specific gene codes some informations. So the mutation in these genes leads to cancer. So what are the major genes involved in cancer development? There are two genes, tumor suppressor genes, oncogenes and tumor suppressors. The role of tumor suppressor is to stop the cancer growth, whereas the oncogene is enhancing the tumor growth. So what happens when a patient has a mutation in tumor suppressor and oncogene? So the cancer stop genes will be deactivated, means they don't work anymore. So then the cells will be out of control. So what will happen once the cells are not under control? So they will try to find ways to keep themselves alive for a long time. This is called cancer. So what these cancer causing genes do? What is the main function of these genes? These cancer driver genes, that is the oncogene and the tumor suppressor genes. So they basically tell the cells to not to listen to the normal signals, normal controls. They completely act independently, and the main motivation of this cancer cell is to find ways, means the way refers to the nutrients, glucose, amino acids, vitamins, minerals. They want more, and they manipulate their structures to get more nutrients, and they grow faster, they divide faster, and finally they form a cancer. So what are the modern foods that can cause cancer now? So as I said, high energy food, high salt containing food, high sugar containing foods, and preserved foods, frozen high fat diets. These are the major contributors of cancer development. Like all normal cells, cancer cells also need nutrients to grow. So, but The difference is cancer cells have highest requirement for their growth. Normal cells require a certain amount of nutrients. Nutrients means glucose or sugars or amino acids, vitamins. They require a very limited amount, but tumor cells require 10 times or even more, 100 times more than the normal cells, which means you cannot satisfy the tumor cells will not be satisfied with limited amount of nutrients. They need to eat acquire more nutrients for their growth. This is the difference. So now our main goal is to how to stop the tumor cells in obtaining or getting more nutrients. This is the major research that we are doing. So we are finding the mechanisms how to stop tumor cell growth by blocking the nutrients, blocking the food to a cancer cells. In this way, you can slow down the growth of tumor, or you can also try to stop the tumor growth. So how high-fat diet may cause cancer? The reason is high-fat diet induces inflammation, leads to inflammation. So high-fat diet has high energy. So there are, as a consequence of highest nutrition, they increases the inflammation. Inflammation is one of the major reasons for cancer. So all this research strongly suggests that avoiding high-fat diet and preserved food, artificial sweeteners, and preserved fruits, juices are very bad for health, and not only bad, they cause severe diseases, cancer, and other diseases like diabetes and heart disease, kidney disease. So we have to be cautious cautious with the preserved foods. So we should always prefer to eat healthy and fresh foods. That is more important for better life and disease-free life for a long time. So why nutrients are important? Why nutrients are important um, for the normal healthy life? Because eating fruits and vegetables are more important for avoiding dangerous disease like cancers. According to new research, people who eat high amount of fruits, vegetables every day have a lower risk of cancer as compared to people who eat less vegetables and less vegetables and fruits. So these findings, they strongly suggest that eating fruits and vegetables is more important to avoid cancer because these vegetables and fruits has a lot of medicinal properties, like because they have phytochemicals. These phytochemicals, they fight against cancer, and they prevent the abnormal tissue formation, that is cancer formation. That's why the fresh fruits and vegetables play an important role in maintaining normal healthy life. So our topic is mainly to be cautious and to avoid high-fat diet and to prevent the cancer. So in this way, we can prevent the cancer. And besides, apart from nutrients, we can also try to improve the immunity. And by boosting the immunity, we can also overcome cancer. How we can overcome cancer by improving immunity? So immunity can be boosted by doing routine exercises and sports, and avoid alcohols, and avoid using processed meats and artificial sweeteners, and routine medical diagnosis. For example, women after the fertile age, they should undergo cancer diagnosis frequently, at least every 3 years once. The women, so they should undergo some diagnosis. For example, cervical cancer diagnosis, ovarian cancer diagnosis, endometrial cancer diagnosis. These are common cancers. Women are at high risk after during their fertile age. They should do this test to prevent their life and to for their long life. And also the diet is, as I mentioned earlier, the diet is also very important for healthy life and without cancer. So to conclude my talk, the cancer is not all the cancers are curable, but they are preventable by maintaining healthy lifestyle. So this is the conclusion of my talk. And finally, I would like to thank the committee members for giving this great opportunity. I wish the Knowledge Fest for the great success. Thank you again.